All right, so we've almost finished with the central dogma. We've seen this process by which messenger RNA is made from the DNA, and that process is transcription. And we've seen then how the messenger RNA is read by the ribosomes to make protein. That process is translation. We've seen the steps that occur between transcription and translation. And now the question is this. We go DNA, RNA, protein, information flows in that way, and then that ends up determining the trait or the phenotype. So that's our last step, is to look at that. Problem is I can't give you a general theory of this because there's so many special cases that it really is, there's no general theory that will express everything that happens, except for the idea that proteins produce the compounds. They're the enzymes that help produce those compounds. They are the structural units of things in the cell. The cell determines what tissues can do. Tissues determine what organs can do. Organs determine what organ systems do. Organ systems determine what the organism can do, and so on. So that's the hierarchy leading up towards phenotype. So I want to make that a little bit more precise, though. It's easier to sort of see it if you can see a single example. And that example that I'm going to use is this example of sickle cell anemia, which is a particular disease of the blood. It's a genetic disease, and it tends to occur mainly in people who have descended from people who evolved in a region that had to deal with a particular type of malaria, in particular the falciparum form of malaria, which is the deadly form, what we call the malignant form. So sickle cell anemia is this. If you look here, this is just a nice little electron micrograph of red blood cells. This picture, by the way, is actually just false color, but they're making it red so you can see it. If you look at these cells, you'll see these cells are very misshapen. And the reason they're misshapen is because the hemoglobin inside of them has a property that allows them to polymerize. The different hemoglobin tetramers that I showed you before, the two alphas, the two betas, will bond to another one, bond to another one, bond to another one, and make this big long string in a chain that makes the cell, especially when you get it outside the body, sickle up like this. That's why it's called sickle cell anemia, because it's shaped like a sickle. But also it makes it very, very rigid, and so when it goes into the spleen, it can't bend properly through the very, very tiny capillaries in the spleen. And old blood cells have that property that they don't bend very well, and so the spleen knows that if a cell is not bending well and can't get through those little tiny capillaries, it destroys it because it's an old cell. And the problem is these cells aren't old and they get stuck in the spleen destroys them, and that's what leads to the anemia. So the cause of the problem is really exceedingly simple, a very, very simple change in the DNA sequence. If we look here, this is showing the normal codons of the beta globin gene, the gene that we've been talking about. And you see here, this codon right here, the GAG codon, codes for the amino acid glutamic acid. One tiny little change right there, that TA base pair is changed to an AT base pair in some individuals anyway who have this sickle cell trait, and that changes the glutamic acid to a valine. And that valine is what allows the protein to change. Its tertiary structure changes enough that it'll then bind to other hemoglobins, and that's what forms that polymerization that we mentioned earlier. So this is the point, is that this whole disease is caused by just that change. A TA goes to an AT. That's it. So that's the kind of thing I'm talking about. When there's a change in the protein, that changes the trait, and the traits then are determined by how these proteins function. Now, this isn't the only kind of mistake that can happen. It isn't just that these things can change this way. There's all kinds of ways that the DNA can change. I want to go through some of these. Now, I'm going to throw them at you pretty quickly, so it's going to be a lot of stuff. But at the end, I'm going to summarize and try to give it an organization that will help you remember what's going on. But this particular type of change that we have is called a mutation because, again, remember, the mutation is by definition a permanent change of the DNA sequence. And this is a permanent change. This codon went from GAG to GTG. But here's another example of one. If we have this particular sequence, and again, we're reading from 5' to 3', GCT, ATA, blah, blah, blah. If this A right here were to change to a T, then we would have what's called a substitution. In particular, we call it a point substitution. When we talk about point mutations, we generally are referring to mutations that involve only one nucleotide. And again, we see a change, permanent change in this DNA sequence. So this is a common sort of thing that can happen, and that's exactly what this type of mutation is. It's a point substitution in particular, point substitution. But there are other things that can happen. For example, this. If we look at this sequence right here, CAT, 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 a bunch of these goes from histidine, 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 histidine. If we change one of these so that they remove it, this A right here is removed, and so instead of reading CAT, it reads CTC and so forth. Notice how this sequence of codons goes from CAT, CAT, CAT to CTC, and it goes from a bunch of histidines to histidine and then a bunch of leucines, or leucine and then a bunch of isoleucines. So this is the kind of mutation that often occurs, and this is what leads to a very massive change in the protein. And in particular, I'll show you later, this is referred to as a frame shift. So this will completely change the protein in a much more drastic way than this point substitution. This point substitution generally tends to change one amino acid or no amino acids. This will change a bunch of amino acids following the actual deletion point. So of course there's other things that can happen. For example, if we look here, the same exact sequence we started with, a bunch of CATs, so therefore a bunch of histidines. If we insert an A right here, then this reads now no longer CAT, 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 CAT. It reads CAT, 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 ACA, TCA, TCA, TCA. And so we go from a bunch of histidines to histidine, then threonine, and a bunch of serines. Again, this type of mutation is a point mutation because it involves only a single nucleotide, but in this case it's inserted into the DNA sequence, and so we call it a point insertion. So those are the three that we've seen: the point substitution, point deletion, point insertion. Notice that the point deletions and point insertions have a much more drastic effect on the primary structure of the protein than just simply usually a single substitution. So here's why it has such a huge effect. It changes the primary structure. See, these are a bunch of histidines, and then we now have histidines, and threonine, and a bunch of serines. Those changes will change the tertiary structure of the protein, the folding, that big blob that it turns into, and that changes its function. That's the key thing to understand, and I mentioned this at the very beginning of the course. A protein's function is determined by its tertiary structure. Its tertiary structure is determined by its primary structure. And the primary structure, therefore, is critical in understanding how proteins work. It determines not only what sequence of amino acids it is, but how it folds not just in the tertiary structure, but also into the secondary structure, in the quaternary structure, and all the structural hierarchical units of proteins. So these kinds of things leading to these big changes, which I'll call frame shifts, are massive, usually have much, much bigger effects than simple point substitutions. But that's not the only thing that can happen. You can also have these things that are called repeat expansions. So for example, you can have CAT-TCA, and this sequence here is the normal sequence that we would find in a DNA sequence. And you see right here CAG, which codes for this amino acid, glycine, can be repeated, CAG, CAG, CAG. It makes a series of them, and that occurs when the DNA is being duplicated, and it's a mistake that occurs through a mechanism which I haven't talked about. But you can take these codons and repeat them over and over again, and you get these repeated sequences of codons that will then repeat this glycine. Normally you should only have one glycine there, now we've got three. So that's called repeat expansion. It's not really a point mutation. It's, however, different than the next level of mutation. The next level of mutation actually involves usually a chunk of the chromosome. It can be anywhere between 100 or so nucleotides, or thousands or even millions of nucleotides. For example, we can have a point where an enormous section of the chromosome is completely deleted. So right here we have one chromosome right there, and this area that's kind of brownish is gone after the chromosome is replicated. So that's referred to as a chromosomal deletion. It's not it's not involving one single nucleotide, it's involving an entire section of the chromosome. And in this particular case, it would be hundreds of thousands to millions of nucleotides that are involved here. So we can have an enormous change there. Now this is bigger than just a frame shift. Here we're removing entire genes and gene cassettes and whole regions of the chromosome. So this can have a very, very massive, drastic effect on the phenotype. And you can also have this, where a chromosome, for example, has a section of it that's duplicated and then reinserted right next to it. So you see this sort of brown region here gets copied, and then it gets inserted right here. So this is what's called a chromosomal duplication. So we're duplicating this and inserting the new section here. And that leads to another type of chromosomal mutation called a chromosomal insertion. There are other insertions that can occur that again involve sections of the chromosome that are anywhere between a few nucleotides to millions of nucleotides long. Other things that can happen are this. For example, you have a translocation, and this is a very famous type of mutation that is important in a type of leukemia. You can have, for example, a section of one chromosome and a section of another chromosome swap. So here we have this section of a chromosome number 4 and this section of chromosome number 20 swap, so that this bottom section on the long arm of 20 ends up where this bottom section on the long arm of chromosome 4 is, and so forth, and vice versa. So this goes here to make this, and this green goes here to make this. That kind of translocation is an event that does occur and has a very important clinical significance in a number of diseases, and again, it has a huge impact. And in particular, it can fuse genes. And this particular example that I'm talking about, this type of leukemia, it fuses one gene called BCR and the other one called ABL, and it fuses them together and makes a functional product that actually is pathological. And then there's another example here, another type of chromosomal change, where you can have, notice, look at the banding pattern. There's a dark blue, a green, big blue, big green, and a small blue, tiny green, small blue, tiny green. And you see it switches so that now the tiny green, small blue, tiny green, small blue, big green, big blues are upside down. That's referred to as an inversion, and that can happen again on a big section of the chromosome that just basically switches and flips like that. So these are all different types of mutations that we see. This is not even an exhaustive list of the types of mutations that we see, but they can cause huge changes in the phenotypic expression of these genes. So the consequences that arise from these mutations can also be classified. So these are not the types of mutations that I'm talking about. These are the consequences of those mutations. And what they're showing, notice up here, is the messenger RNA and the ribosome reading that messenger RNA gene and making the protein, showing you the central dogma and the step of translation. So consider this. Look at this. So here we have a normal amino acid sequence here that's made from this messenger RNA sequence. And again, we talked about this before. The transfer RNA carries these anticodons. The transfer RNA carrying the anticodon CGA carries an alanine, and it binds to the codon GCU, and that is then the sequence alanine, then isoleucine, arginine, leucine, glycine, tyrosine, so on. That sequence then will be long enough. Notice these dots. It means it's going to go on for quite some way. And that sequence of amino acids is long enough to fold into this tertiary structure and makes our protein. So that's the normal thing, and this protein would be an enzyme or some other structural unit or something that causes the cell to perform in a particular way. Now, if we look here, look at this GGU, that particular codon which codes for glycine. Now notice right here what's happened in this sequence is the GGU has become GCU. In that particular type of mutation, what is it? What kind of mutation is that? If we go back up here, you'll notice that that is a substitution. The G's been substituted with a C. So that changes, however, the glycine into an alanine. Everything else is exactly the same. See, on this side, tyrosine, serine, alanine, cysteine, tyrosine, serine, alanine, cysteine, so forth. The same side over here, alanine, isoleucine, arginine, leucine are all the same. The only change is here. Whenever that happens, whenever you have a change in the amino acid, we call it a missense mutation. Means that the sense has changed. The actual translational meaning has changed. Okay, now consider this one, though. Here's another example. In this case, this particular sequence right here is UGC UAA, has changed then to, it looks like UAA here. And right after the cysteine, it should be AUA, but instead it's UAA. Okay, now this is not exactly quite correct. It looks like those two things switched. But at any rate, this change here causes then an early stop codon. And so instead of reading all the way through this cysteine and continuing on to isoleucine, histine, valine, alanine, and so on, it stops. It's a stop codon. So what happens then, the cell doesn't know that that's a mutation, so the cell does this. The ribosome binds to it, it goes to it, and sees that as a stop codon, and it treats it like a stop codon. The termination factor is going to bind to this. It's going to knock the ribosome off, and the whole protein stops being built at that point. So you end up with a much truncated protein. The body, the part towards the carboxyl end is gone, and this then ends up completely changing its function. That type of mutation where you end up with an early stop codon we call a nonsense mutation. So don't get that confused. Don't use the word nonsense in this context as you do in normal everyday speech. It doesn't mean that it's not meaningful. I mean, it sort of means that. It means that, though, that the protein is truncated early. Okay, so those are two different types of consequences that can occur from these point mutations. You can get a nonsense mutation where you change a normal sequence into a stop sequence. You can get a missense mutation where you change a normal sequence into a new amino acid. So here's the example. TTC makes lysine. And here, this right there, T has been mutated to a C, and now that codes for arginine. And so this is what we call a conservative missense mutation because this particular type of amino acid is the same as this type of amino acid, has the same kind of side chain. Non-conservative means that the side chain is different. So remember, the side chains can be polar, ionic, or nonpolar. If it goes from polar to polar or nonpolar, whatever, we call it conservative. If it goes from nonpolar to polar, nonpolar to ionic, or ionic to nonpolar, whatever, we call that non-conservative. There's also this possibility. Notice TTC in this case, in this example, has been changed to TTT. The final T has experienced a point substitution from C to T. In that case, if you look at the dictionary, you'll see that TT anything, doesn't matter what the third codon position holds, anything that starts with TT is lysine. So TTC and TTT are both coding for lysine. It's a mutation, but it has no effect, at least not a major effect. It can have a slight effect sometimes, but in this case we call it silent because it doesn't change the translation of the protein at all. Okay, so then the final consequence is one that we've actually already seen. In this case, look at this sequence right here, CAT, TCA, CAC, pardon me, codes for the sequence histidine, serine, histidine, valine, leucine, methionine, leucine. Okay, so that sequence right here is the normal one. If we move our reading over exactly one for whatever reason, instead of starting our codon with this C, we start the codon with this A, then we have ATT, which codes for isoleucine, and then CAC, which codes for histidine, and then threonine, and tyrosine, and serine, and cysteine. And notice that all of these have changed. All the amino acids have changed simply because we're starting a different spot. We're starting our codons at a different spot. That's called a frame shift. And the reason for that is that if you look at this sequence right here and we say, okay, here's a codon CAT, the next codon TCA, the next codon CAC, and so on, we call that a reading frame. What the reading frame is telling us is, where do we start and stop codons? If we move this over slightly, then we're starting and stopping codons in different spots. And those different spots are simply defined by where it is we've decided what our codon is going to be. So this is a different reading frame. It's the exact same DNA sequence, but it's a different reading frame because we're starting and stopping our codons in different spots. So now let's go back to this example we talked about earlier, the point deletion. Notice right here we took out an A, and that A was deleted, and that then made this codon, CAT, read now CTC. See, that's CTC. So we've now removed that, changed this codon, and every other codon downstream is now in a different reading frame. So that is what we call a frame shift. The same thing happens then with a point substitution. If we insert an A right here, this normal sequence CAT, CAT becomes CAT, but now we have a new A in there, and the new codon is ACA, and right there is the break. So after that, the breaks occur every 3 because, again, the DNA is really stupid. It just always has 3 length 3 nucleotide codons, and this changes the reading frame again. So again, we have an example of a frame shift. Okay, so that's just a partial list and description of the consequences of a variety of different kinds of mutations that you're going to see. The reason I need you to know all of those terms is that you're going to very soon be reading primary literature, and you need to know what they're talking about because this is how they're going to refer to these changes. And it's a good idea to start to get a sense for how to organize your thoughts about these things. And the way this works is there's 2 different ways I've been looking at this. One are the types of mutations, and the other is the consequences of those mutations. The types of mutations we talked about included 3 different types. Point mutations, which involve only one nucleotide, this sort of intermediate one, which involves a number of nucleotides, and then chromosomal changes, which involve a large chunk, anywhere again between maybe tens of nucleotides to thousands to millions of nucleotides. The point mutations include substitutions, deletions, and insertions, changing one nucleotide for another, deleting one out of the sequence, or inserting one into the sequence. This intermediate level of types of mutations includes those repeat expansions, where a single codon is repeated over and over again. And then finally we have the chromosomal changes that include deletions, duplications, which also includes a chromosomal insertion, translocations, where one part of the chromosome binds to another, switches with another part of the chromosome, and then the inversion, where a section of the chromosome is essentially removed, flipped, put back in consequence. So those are the mutation types, and they lead to these different types of consequences. You can have silent mutations where the mutation has no effect on the amino acid sequence. You can have missense mutations where the consequence is a change in the type of amino acid that's being coded for. You can have a nonsense mutation where you have an early stop codon where the stop codon should not be, and then you can have a frame shift, any sort of change that causes the DNA to be read in a different reading frame. So what you should try to do is to get examples, and there's a number of examples in your book, to get a sense for being able to identify these types of mutations. That's how I'm going to test you on it. So on the quiz you'll see, I'll give you a sequence, tell you what the new sequence is changed to, and then you'll have to figure out, first of all, what type of mutation it is. Is it point, chromosomal, repeat expansion, what type of point, what type of chromosomal, whatever, and what its consequence would be: silent, missense, nonsense, frame shift, or complete deletion of the gene.